Okay, so now we need to delve into the last portion of the external features of the bacterial cell, and this is where we will look at the cell wall. Now, we have studied and we understand that all cells have a cell membrane, all cells, animal, plant, you name it. However, not all cells have a cell wall. Bacterial cells will typically have a cell wall, and they're a little different from plant cells because plant cells also have a cell wall. Animal cells do not have a cell wall, and the specialized structure that we're going to have is called peptidoglycan. So primarily, the cell wall is composed of a lattice-like structure of carbohydrates, that's the glyco part, and proteins, that's the peptide part. So it is proteins and sugars, or carbohydrates, that make up the structure that will make up the cell wall of a bacterial cell. Now, let's take a look at the difference, because there's going to be two types of cell walls. In bacterial cells. So notice I also said in bacterial cells. Okay? The two types, one of which is known as gram-positive, and the other which is known as gram-negative. Now, they have a really good example of those two cell types on page 68. So we're going to go ahead and pull up our page 68 and take a look at the differences between the two. The top one is your gram-positive cell wall. Take me just a second. There we go. Okay, so our top one is the gram-positive cell wall. You'll notice here's your layer of peptidoglycan. Very thick layer. So we'll remember the difference between gram-positive and gram-negative. Gram-positive has a thicker peptidoglycan layer. Now this gray here, this is going to be teichoic acid going partway through and lipoteichoic acid. So those are going to be the structures that go through your cell wall, helping anchor it in place and things of that nature. So teichoic acid is going to be present in your cell wall, as you notice here. Very thick peptidoglycan layer, and then here's our cell membrane here. Okay? Now let's take a look at our gram-negative layer. Totally different. There's this tiny little layer here in the middle. That is your layer of peptidoglycan. So we'll have an outer membrane that goes over the peptidoglycan. Let me zoom in on this just a little. There we go. Okay, so let's zoom in on the gram-negative and we'll take a look at it. So this is our gram-negative cell wall. Let me zoom out. Whoop. And then zoom in on our gram-positive. There's our gram-positive. Notice here's our cell wall. There is the peptidoglycan layer with the teichoic acid going through it. There's our regular cell membrane that's going to be on the outside of our cell. And this is on the outside of that. Okay? All right. Now let's take a look here. That is our gram-negative. So we just looked at the gram-positive. Now we're going to look at the gram-negative. Very thin layer of peptidoglycan right here. That is it. See where it says peptidoglycan? Yeah, where is it? There it is. Peptidoglycan layer of cell wall. Right there. Now, we're going to have a membrane that's normal. And then we're going to have an outer membrane that goes around the outside of our cell wall. Between these two membranes, the peptidoglycan sets in an area known as the periplasmic space. See it right there, peri meaning between, plasma, okay, space. So this is going to be the periplasmic space where the peptidoglycan sits between these two membranes. Now let's take a look at the outer membrane of the cell wall on a gram-negative cell and look at some of the specialized things it has. Lipopolysaccharides. Some of them will contain what we call lipid A. Lipid A is going to secrete toxins that are very dangerous to the host that has the bacteria. It can be a very dangerous side effect from that pathogen. So there are the LPS, you'll see it abbreviated a lot of times, lipopolysaccharides, which is a fat with a sugar attached that contains lipid A, which is a specific lipid or fat. All right, sometimes you'll have porins through them, which is very normal, things of that nature. I have a really cool diagram at the office. I'll pull that out and also see if we can see pictures of this, and I'll do some explanation on cell walls. Unfortunately, you'll probably have to just go ahead and take your quiz and read the lectures and go through the lecture, and then we'll be able to do another lecture on that because I don't have that in front of me. So there's your cell wall, gram-positive and gram-negative, and there's a difference between the two. We talked about how the gram-negative rinses off and changes to a second color when we do gram stain. Well, now you can kind of see the understanding of why, because look how thin the cell wall is versus your gram-positive up here and how thick it is. So this layer is much, much thicker, and it's harder for that to lose stain. So it's going to stain the primary color, and then this one will stain the secondary color when we go back and stain it again. Let's talk about some exceptions to the rules. And we got a couple. All right, one of which exception to the rule is a gram-positive bacterium that we don't call gram-positive because it is a little different. Okay, and I'm looking for it. It's on page 67 right in here. So they're talking about Mycobacterium. There it is right there. Tuberculosis and leprosy would be Mycobacterium, types of them. Mycobacterium tuberculosis, Mycobacterium leprae are both the disease-causing agents for those specific diseases. They have walls that instead of having teichoic acid, they have what's called mycolic acid. And it's a very waxy lipid. Well, if we understand much about wax, we understand that wax will basically roll right off of any sort of, or water will roll off of wax, right? So most of our dyes that we use, or all the dyes that we've been using or will use in class, are water-based dyes. Okay, they may be acidic, they may be basic, but they're both water-based dyes. So therefore, we have to have a special system to be able to allow us to stain these. And we stain these specific organisms with what we call an acid-fast stain. Okay, and that's what an acid-fast stain is designed for, something that has a more sensitive cell wall that will resist the actual stain. Now, why do they have this? Well, this also helps them with not allowing them to dry out, so it prevents them from desiccation. That's what we call drying out. Makes them difficult to stain. So that's why we use our acid-fast, and we also call them acid-fast bacteria just because of that. All right, so we have another type of bacteria, another exception to the rule, that does not have a cell wall at all. Let me back out of here. There we go. Bacteria without cell walls. And that talks about it in page 69. This is mycoplasmas that do not have cell walls at all. They lack them entirely. So we at first thought they were viruses because they're very, very small and they lack a cell wall. So we thought, okay, they must be a virus. But then we learned that they are definitely prokaryotic. They have specific set structures such as DNA and the specialized ribosome for bacteria. So we understand that they are definitely bacteria. They just lack a cell wall. So we have to put them in a special category because they don't fit with the others. All right, so that gets through our cell walls. Hopefully that wasn't too much and too in-depth. Hopefully it was fairly simple. And then we're going to go into our structures that are internal of our cells.